So part 3 is more PYMOL practice, continuing what you did last night. So we're going to reopen our session. It should be an open recent, and I've saved it here as PYMOL session, so I'm going to reopen that. And as you can see, we've still got my symbol display with my protein's backbone. And then we've got our heme group here. We're going to practice changing the color scheme, and we're going to add the primary sequence information. So what we want to do is we want to color by chain, and we're going to use the feature called chain bows, and you'll see why I use this feature in just a moment. What I want to do next is I want to go to the program menu at the top of my screen. This is the program menu at the top of my screen. And I want to go to Display, Sequence Mode. And I want to, first I'm going to display the whole sequence. And as you can see, it displays this sequence of amino acids, but it does it with single letter codes. And we've been working with 3-letter codes. So I'm going to make sure that I use 3-letter codes. So I'm going to go back to Sequence Mode. And I'm going to choose Residue Names. And so now you can see that we've laid out our amino acid sequence by their 3-letter codes. And if you scroll over, you can see every amino acid that is present in your molecule. This is important. You can also see the heme group, and if you click on it, it selects the heme. You can deselect it if you want to select a specific amino acid. Here's serine. Note that serine is at the end of our molecule. I'm going to deselect that. I'm going to go back over here. And glycine is at the other end of our molecule. So what benefit does chain bows show us? You'll notice that it always is going to start with a blue amino acid. This is glycine over here. And it's always going to end with a red amino acid. This is serine on the other side. So now we can see sort of both ends of our molecule, serine and glycine. And the red end of the protein chain is going to indicate either the N terminus or the C terminus. How can you tell? Well, we know that glycine is the start of the protein, and we know that serine is the end of the protein, which means that glycine is showing the N terminus, the amino terminus, and serine is showing the C terminus, or the carboxyl terminus. So we want to know which residue is number 99 next. So I'm going to deselect these two, and I'm going to scroll along until I get close to 99. So here's 96, 97, 98, 99. I click on 99, and it pulls up lysine. You'll notice that, lysine has a big R group that comes off of the main chain, and then it has some, its normal amino acid structure attached to the normal chain. Is it close to the heme group or far away? It's fairly close. It's pretty close to the heme group. Next, we'll try another representation in a different color scheme. We're going to Action. We're going to a preset. And we're going to use ball and stick. Aha. And then we see all of our atoms here, but they're not colored specifically to each atom. So instead, I am going to color them by element. I'm going to go to Color, by Element. And as we normally do in science, I'm going to use gray, this color gray here, for my carbons. Now we can see all of my carbons are colored in gray. Obviously, my oxygens are red. My nitrogens are blue. And I also see some yellow, right? And we could click on the yellow if we wanted to know exactly what that atom is. Here's one right here. And this is S, so this is a sulfur. What parts of the structure are shown in ball and stick and what were hidden in the simple view? What are the drawbacks or limitations of the ball and stick view? What are the benefits to coloring by element over coloring by chain bows, especially in this view? We're going to try a new action. We're going to go to action preset technical. Ooh, and we have a very different looking molecule here. As you can see, the heme group is still in the middle. But what do you notice? Well, we've got a, maybe we should click on a lysine again, so we can go observe lysine. Here it is on the side. We can zoom in a little bit. We can see that every atom in lysine is now not showing other than the connection of two lines. So we are showing the bonds, the specific bonds, right? So here's our carboxyl group. Here's our side chain over here. One thing that we should note in technical preset is that we see these. We see these dotted yellow lines. What do you think those dotted yellow lines are representing? It's one thing to think about. What unique feature of a protein is probably being shown by those dotted yellow lines? So what is the benefit of having a technical preset? I'm going to go back to center. And finally, we're going to use a space filling representation. So I'm going to go to show. And I'm going to show spheres. Boom. Wow. Okay. So every single atom in our molecule is now showing up as spheres. And this would be sort of the electron cloud that is taken up by each individual atom. And you'll see where, oxygens are bonded to carbons. Those atoms are sharing an electron cloud, which makes sense based on what we know from chemistry. I'm going to go back to coloring it by element. So we can see sort of where our carbons are, where our oxygens are. You can even see some sulfur in there as well. What are the benefits to this? We want to hide some of the hydrogens so that we're not showing too many. So I'm going to go to hide. I'm going to hydrogens, and I'm going to select nonpolar. So most of our hydrogens are involved in some sort of polar connections. And that's it. We've finished our brief introduction. I hope you were able to take the images that you needed. Now we'll start examining protein structure in more detail.